Today, we'll be discussing genetic and molecular dissection of somatic embryogenesis. Somatic embryogenesis occurs naturally in various plant species and is widely used for clonal propagation, transformation, and regeneration of different crops. Somatic embryogenesis shares some developmental and physiological similarities with zygotic embryogenesis, as it involves common factors of hormonal, transcriptional, developmental, and epigenetic control. Somatic embryogenesis relies on the totipotency of the plant cells, that is, their capacity to de-differentiate and differentiate in a new cell type. The first stage of somatic embryogenesis are characterized by the induction of the expression of numerous stress-related genes. Somatic embryos can be induced directly from in vitro cultivated plant tissues on a low oxygen medium, or indirectly by cultivating embryogenic tissues such as callus on oxygen-rich medium, leading to the transition of somatic embryos by switching to a low oxygen medium. The genetic and molecular dissection of somatic embryogenesis involves identifying genes, analyzing the transcriptome and proteome to identify genes involved in somatic embryogenesis, then identifying genetic loci that regulate the transition from embryogenesis to germination, mapping the signaling pathways involved in the induction and regulation of somatic embryogenesis, and identifying how epigenetic regulation activates or rechannels developmental cell memory. First is transcriptional control of somatic embryogenesis. The main way to regulate gene expression is the transcriptional enunciation through transcription factors. In Arabidopsis, the ectopic expression of some transcription factors such as leafy cotyledon genes, baby boom, then Wuerthel or Wuerthel-related homeobox genes, or Agamous-like 15, means AGL15, can increase the efficiency of somatic embryogenesis induction and lead to the formation of somatic embryos without adding hormones. Several of the transcription factors are involved in hormonal signaling, while others control cellular differentiation and organogenesis. First is LAFL. Here we'll discuss the role of LEC1, LEC2, ABI3, and FUS3. Abscisic acid in sensitive 3, ABI3, then FUS3 and leafy cotyledon 2, LEC2, are transcription factors forming the AFL group. They belong to the plant specific B3 family of transcription factors, characterized by a highly conserved B3 DNA binding domain of approximately 110 amino acid residues, first characterized in ABI3 or VP1. B3 containing factors can recognize specific target DNA with 5-GATC to 3- sequence known as RY boxes and conserved within the promoters of storage genes. Together with leafy cotyledon 1, LEC1, are NFYB transcription factors involved in CAAT box complexes. AFL form the LAFL group of master regulators of seed development. The expression of LAFL genes is regulated by transcription factors such as BBM, hormonal signaling, and chromatin modifications. AGL15 is involved in hormonal signaling and control the embryogenic induction. IAA30, a main actor of auxin signaling and perception, or LOP40, a transcription factor putatively involved in the formation of organic boundaries and gibberellic signaling. LEC2 and AGL15 control each other in a feedback regulatory loop. LEC2 is also known to rapidly induce the expression of auxin-related genes such as IAA1, IAA17, and ACS4, and key enzymes involved in auxin biosynthesis such as YUC genes YUC1, 2, 4, and 10. Among these auxin genes, only YUC4 is direct target of LEC2. Next is baby boom. Baby boom, also known as Pathora4, PLT4, a transcription factor belonging to AP2 transcription family, characterized by DNA binding domain of 70 amino acid residues that were first characterized in APETALA2, means AP2 and ethylene responsive element binding proteins. The AP2 family is divided into two subfamilies: transcription factors. First is transcription factors that count one AP2 DNA binding domain and are generally involved in response to abiotic and environmental stresses. And second is those counting to two DNA binding domains known to regulate developmental processes. BBM contains two AP2 or ERF domains and is a member of um, Antigmenta-like subfamily, means AIL subfamily, composed of 8 members, all carrying specific functions in the division of meristematic and embryonic cells, involved in the regulation of somatic embryogenesis and its induction when they are ectopically expressed. BBM is expressed in zygotic embryos and regulates cell identity and the growth of the root meristem. LAFL genes are upregulated after BBM induction and together with the AGL15 are BBM positive regulator during the somatic embryogenesis processes and are key components of the BBM signaling pathway. Next is virtual and virtual related homeobox genes. WUS is a homeobox transcription factor that is described as a key regulator of meristematic stem cell fate. It also performs an essential role in somatic embryogenesis by promoting the vegetative to embryonic transition and the maintenance of embryonic cell identity. WUS is involved in regulation of the embryogenic cell, means totipotency, and meristematic cell fate. In Arabidopsis, WUS mutants can form somatic embryos, although seedling display the same phenotype as seed-derived WUS plantlet. The overexpression of WUS and gain of function mutation can increase somatic embryo production or induce the formation of somatic embryos from vegetative tissues without addition of external hormones. WUS can also be used as a marker gene of dedifferentiation after somatic embryogenesis induction in some other plants. Next is Agamus-like 15, AGL 15. Agamus-like 15 is a transcription factor belonging to the MADS domain protein family. These transcription factors are known to share a specific MADS DNA binding domain containing a 55 to 60 amino acid sequence conserved among eukaryotes. AGL15 can target and regulate the expression of numerous hormone-related genes, including gibberellic acid and ethylene metabolism, but also auxin signaling via GA3OX6 and IAA30 activation. AGL15 activates its own expression and regulates LEC2, ABI3, then FUS3 in a positive feedback regulatory loop. The ectopic expression of AGL15 in Arabidopsis thaliana and in soybean increases somatic embryogenesis capacity. Next is wound-induced dedifferentiation 1, WIN1. WIN1 means WIND1 is an AP2 or ERF transcription factor with two AP2 domains involved in callus formation in response to wounding and performs a critical role in the acquisition of regeneration competency and regulating cytokinin signaling. The over and ectopic expression of WIND1 leads to the callus formation on explants without induction through the addition of hormones in the culture medium or wounding. WIND1 not directly involved in induction of somatic embryogenesis, but in the induction of callus formation and regeneration competency. Next is epigenetic regulation of somatic embryogenesis. Epigenetic is the study of how environmental and behavioral factors can alter gene expression without changing the DNA sequence. Right? DNA methylation profile and chromatin accessibility are constantly changing in response to cellular needs and environmental constraints. The different abiotic stress to which plants are subjected can either increase or reduce DNA methylation depending on the type of stress, the plant species, or even the organ considered. It should be noted that epigenetic changes induced can be transient or stable over a period of time and can also be transmitted to the offspring. Then it can easily be hypothesized that epigenetic modification correlate with somatic embryogenesis process. In particular, the efficiency of cellular differentiation is linked to the methylation profile of DNA. DNA methylation level in callus decreases during embryogenesis, and the important change observed in transcript level during callus induction and somatic embryogenesis. It reflects the epigenetic reprogramming. Then polycomb repressive complexes. PRC1 and 2 are required for establishing and maintaining a stable epigenetic repression in response to developmental or environmental signals. Briefly, PRC2 has a histone 3 lysine 27 trimethylation activity, and PRC1 can recognize H3K27 Me3 and lead to chromatin compaction through histone H2A lysine ubiquitination. PCR2 is the main factor involved in cell identity to maintain a stable repression of embryogenesis program genes. PRC1 and PRC2 can repress the expression of several genes involved in cellular differentiation and callus development, as well as somatic embryo formation, including WX5, WX8, AGL15, LEC1, LEC2, ABI3, FUS3, BBM, PIN1. Next is hormonal control of somatic embryogenesis. Multiple genes in the plant life cycle, such as developmental or responses to biotic and abiotic stresses, are controlled or regulated by hormones. Auxins are known to play essential role in all the aspects related to plant development, from cellular division to elongation through cell identity and organogenesis. Auxin polar transport by protein means PIN efflux and AUX influx lead to differential distribution with gradient and maxima that are essential for plant development. During zygotic embryo development, local auxin production and polar transport and gradient are mainly mediated by PIN protein. More specifically, in Rhabdopsis, the expression of PIN1 and PIN7 genes in the apical parts of the young embryo is at the basis of the acquisition of the apical basal orientation in concert with AUX1, LAX1, LAX2, playing a cooperative role. During somatic embryogenesis induction, auxin response factor genes display specific expression being up or down regulated, suggesting that auxin signaling is central to the process. Thank you.